Okay, also welcome everyone from my side. So as mentioned, my name is Chao Sheng, and I am the Global Product Manager for the Regenerative Medicine portfolio at Miltenyi Biotec in Germany. So today, we also have my colleague Felix here, who is an expert in flow cytometry cell sorting technologies. And together, we will introduce you our workflows and our latest technologies for automated closed manufacturing and sorting of pluripotent stem cells and their derivatives for clinical applications. So this is today's agenda. I will first very quickly introduce you our solutions for automated PSC expansion, differentiation, and also standard flow cytometric QC analysis with our widely used CliniMACS Prodigy cell manufacturing platform and the MACSQuant Analyzer. And then Felix will continue to introduce you the gentle and GMP-compliant cell sorting technologies using the MACSQuant TytoCell Sorter. Well, let's first have a look at the general steps of PSC applications. Well, in particular, regenerative medicine. Well, there are many important requirements during the manufacturing process. So, for example, it needs to be GMP compliant, scalable, and reproducible. Meanwhile, well, to really make sure the final cell products are also affordable to the patients and to the healthcare system, the manufacturing process also needs to be cost effective. For all these reasons, the manufacturing PSCs and PSC derivatives for clinical applications are very challenging and complex. Most of the current manufacturing processes actually they involve multiple devices, very skilled and experienced operators, and high-class clean rooms. And also many separated open handling steps are also introduced into these workflows. So at the same time, standard in-process control and quality control assays are required throughout the entire processes. In addition, in certain applications, the targeted cell populations also needs to be isolated in a sterile, gentle, and GMP-compliant environment. All these factors together can really limit the manufacturing skill, also compromise the reproducibility. At the same time, it drive up the manufacturing cost. So everyone in this field is facing the question that how to make these very complex manufacturing processes more standardized, more scalable, and also more affordable for the end users. So here, these are the solutions from Miltenyi Biotec. First, with the CliniMACS Prodigy, then the separated open handling steps, the multiple devices, the skilled operators, and also the high-class clean rooms can be integrated into one closed and automated system. And then the user can also build customized PSC expansion and differentiation processes on the CliniMACS Prodigy. Then, with the MACSQuant Analyzer, standard iPSC QC assays can be realized by automated flow cytometry analysis. Last but not least, the MACSQuant TytoCell Sorter can also be applied to isolate the desired cell populations with a very gentle, fast, and GMP-compliant cell sorting technology. So let's first have a look at the hardware features of the CliniMACS Prodigy. Together with the GMP-compliant and single-use tubing sets, the CliniMACS Prodigy allows automated cell processing from starting materials to the final products. Upon your request, so the CliniMACS Prodigy can automate all these cell processing steps. For example, sample preparation, density gradient centrifugation, cell washing, medium change, magnetic cell separation, cell stimulation, genetic modification until the final product formulation. And based on the CliniMACS Prodigy, so now the adherent cell culture process, with this process, the users can culture their cells in two different units, in the centrical unit or in the external culture vessels. First, in the centrical unit, so there's a CliniMACS Prodigy chamber lies inside. And inside this centric unit, it supports the automated adherent and non-adherent cell cultures. You could automate cell feeding, cell harvesting. You can control the temperature and also the concentration of air and CO2. You can also visualize your cell cultures with the integrated microscope in the centric unit. Meanwhile, there are also multiple input ports for medium, for cytokines, for your cell cultures. Well, if larger surface space is required, by the way, this chamber offers 100 square centimeter surface space. So if larger surface space is required, then the external culture vessels can also be easily connected to the CliniMACS Prodigy tubing set via sterile routing. For example, these multi-layer cell stacks. Then the users can connect, actually we have tested, they can connect maximally 3 of these 5-layer cell stacks at the same time. And this can offer roughly 10,000 square centimeter surface space for the cell culture. Now I would like to shift your attention also to the adherent cell culture software. Well, when we designed this software, actually our goal is to make sure the software is flexible enough to culture different cell types. So that is why we modularize the individual cell handling steps. So now you can find these different modules on the main menu of the adherent cell culture process. So for example, for density gradient centrifugation, for coating, for inoculation, for culture, medium change, and harvest. And then the users can also freely select and combine these different modules. As a result, very complex workflows, for example, directed differentiation of pluripotent stem cells, can be realized on the CliniMACS Prodigy adherent cell culture process. And within each module, you can also set up the cultivation parameters according to to your established workflows or SOPs. For example, in the coating module, the users can set up the coating temperature, the coating time, the volume of the coating reagents, all according to their established SOPs. So these are the hardware and software features of the CliniMACS Prodigy. So now I would also very quickly to show you the MACSQuant Analyzer. Well, we know like a cell manufacturing process also requires very stringent QC analysis. For this, the MACSQuant Analyzer is equipped to meet the demands. So the instrument offers a lot of automated operational features from startup to shut down, including the washing steps. The MACSQuant Analyzer takes on labeling and processing of the samples and standardize the initialization with automated calibration or compensation. So the accurate volumetric cell count can also be performed on the sample itself and does not require any addition of further beads. Then to automate the acquisition and analysis of the iPSC-QC steps, we also offer several on-demand express modes packages. So these are algorithm-based gating tools that can be added to the MACSQuantify software and allow for unbiased, user-independent analysis and resulting in very reproducible flow cytometry data. In addition, well, if you need an express mode for your specific QC application, then we can also customize the express modes to your data analysis. Then, with the Smart Gain technology, you can actually perform multiple instruments alignments for consistent measurements from instruments to instrument, no matter of the operator or where the instrument is placed. In addition, our software can also support or help you with documentation. The MACSQuantify 21 CFR Part 11 software, it delivers everything from audit trial to a good user and role management system, which is necessary to validate the system compliantly. So with this introduction, this information, so now I would very quickly show you some results from our PSC expansion and differentiation workflows. So we have established the workflows on the CliniMACS Prodigy adherent cell culture process for GMP-compliant expansion of human MSCs and PSCs, as well as directed differentiation of PSCs into mid-brain dopaminergic progenitors. Currently, we're also working on a new workflow to differentiate PSCs into cardiomyocytes. but today I will focus on these two workflows in the middle, the expansion of PSCs and the differentiation into MDA progenitors. For the PSC expansion, actually we developed the GMP-compliant iPSC brew GMP medium. And using this medium on the CliniMACS Prodigy, now the users can choose and combine these different modules to automate all the complex handling steps. For example, coating of the culture vessels, PSC inoculation, medium change, cell splitting, harvesting, etc. And we have also performed side-by-side comparisons between manual and the CliniMACS Prodigy-based PSC expansion. And then we use our reaffinity recombinant antibodies for the pluripotency markers, and also our trilineage SNAMAX trilineage differentiation kit to check the trilineage differentiation potentials. So looking at the expression levels of these key pluripotency markers, and also the trilineage differentiation potentials, these two groups, the CliniMACS Prodigy versus manual control, they really gave rise to very comparable results. And on top of this, by using the CliniMACS Prodigy chamber in the centrical unit or the different external culture vessels, the users can also easily reach different PSC expansion scales with the CliniMACS Prodigy. So we know that establishing GMP-compliant PSC banks is a major step in many clinical applications. So for this purpose, the closed automated CliniMACS Prodigy adherent cell culture process, together with the iPSC brew GP medium and our QC tools, could really be very valuable assets for your projects. Well, I mean, not only expansion, the CliniMACS Prodigy adherent cell culture process can also realize very complex PSC differentiation processes. For example, differentiation of PSCs into MDA progenitor cells. So we have collaborated with Dr. Marlene Palmer and Dr. Agnete Kirkby within the EU Neurostem Cell Repair Consortium and have successfully adapted their very efficient manual differentiation processes into the closed and automated CliniMACS Prodigy. So here, this is the process overview, and you can find this also more detailed information in the dedicated application sheets. And here, I just want to show you some side-by-side comparison between manual and the CliniMACS Prodigy. So using the MACSQuant Analyzer, again, the results show that the cells that differentiated on the CliniMACS Prodigy are very comparable to the cells differentiated manually, with more than 80% of the resulting cells co-expressing the key MDA progenitor markers FOXA2 and ODX2, and nearly no expression of the unspecific or the pluripotency markers. In addition to this good differentiation efficiency, the CliniMACS Prodigy also offers automation as well as closed and GMP-compliant environment. Meanwhile, this process, it is also scalable. So in this case, when we started with 1 million iPSCs, and then after 5 days pre-expansion and another 16 days specific differentiation, so we could harvest roughly 3 to 4 billion MDA progenitors, which can be translated to 600 to 800 potential patient doses, considering 5 million cells per cryo unit. The users actually can also further scale it up or scale it down, really just according to their request. So for the end users of the CliniMACS Prodigy adherent cell culture process, we also offer this very comprehensive training program. So this is a glance of the training schedule. So actually it's a 3-day training program, including theoretical sessions, guided and unguided hands-on sessions for the participants, and also the troubleshooting sessions. So this training is actually very helpful for the users to get familiarized with the CliniMACS Prodigy. The CliniMACS Prodigy adherent cell culture process actually only became available on the global market in the end of 2019. But there are already quite a number of labs worldwide started to use it to automate their PSC or MSC workflows. For example, Professor Annelise Benesser-Casali from INSERM, France. So in her presentation at ISCT 2020, She demonstrated that the CliniMACS Prodigy provides a safe, standardized, and scalable manufacturing platform for GMP iPSC master cell banks. This presentation is freely available on our website. So if you're interested, you can also find it and watch the presentation from Professor Annelies Bennesser-Gazelli. Actually, I'm already at the end of my presentation. So if you have any additional questions, we could all either discuss later in the Q&A, or you can also address me via email. Thanks a lot for your attention. So now I would like to give the stage to Felix to talk about GMP-compliant sorting of PSCs and PSC derivatives. Thanks a lot.
Yeah, thank you very much, Chao, for that great first half of our webinar about cell manufacturing, also using the CliniMACS Prodigy Adherent cell culture process. In the second part, I would like to focus a little bit more about the cell sorting aspect of PSCs and their derivatives in a GMP-compliant environment. And before I start talking about this, I just would like to discuss shortly the motivation. Why should you do this? And I think all of us know that the new and innovative cellular therapies are based on very specific cell types you need, or even cellular subsets. And often you start with material which is still heterogeneous, right? We know it specifically also from the immune oncology area, where we see, for example, very heterogeneous cell populations in the starting material if you work with blood, but also in the regenerative medicine field, and when working with, for example, iPSCs, the differentiation efficiencies can vary and might not reach the purities of target cells which are needed for downstream experiments, or also to treat patients. And therefore, you need to purify, to enrich these specific target cells you're interested in, and flow cytometric multiparametric cell sorting is a very good tool to do so. But there's, of course, also a challenge, and that is that the instruments mainly available in the field, so the droplet-based flow cytometric cell sorters, they all are very well capable of purifying cells, also based on multiple parameters using fluorescent markers, but they were predominantly designed for research applications. And that means that most of them are open systems. They do not have, for example, exchangeable tubings or fluidics, so cells will also run through the same tubings, and it's a very complex process to exchange them, if it's possible at all. And in addition, also the sorting conditions can be very harsh and harm the cells so that they won't have a high viability afterwards. And especially if you need functional cells which have to do a job after you transplant them, this for sure is a problem. And here at Miltenyi Biotec, we think we have quite an interesting solution actually to address this, and this is the MACSQuant TytoCell Sorter. You can see the instrument here in the middle of the slide. I'm talking about a multi-parameter flow cytometry-based system equipped with 3 lasers, 8 fluorescent channels, and 4 scatter channels, which already provides quite some flexibility in terms of experimental design and panel design. So the very interesting part also specifically when talking about clinical applications is that the MACSQuant Tyto works with closed cartridges. So this is where the cells go in, and this is where the cells will stay all the time during the sorting process. There are no internal fluidics or internal tubings inside the instrument itself, meaning we don't have to worry about carryover or about cleaning between sorting of different samples. The samples are also never lost inside the system if any problem should occur, because they will always stay within the cartridge and will be accessible from these 3 different ports. And it's also the fact that no droplets or detectable aerosols are produced. And even if we would produce them, we would still be inside that closed cartridge. So we have a very high operator safety, which is also important when working with patient material. All in all, once you set up your panel design, your workspace, and save this, you can reload it, and then it's really a plug and play fashion. So you just scan the cartridge, insert it, press play, and you can walk away and collect your sample, which has been sorted later on. And also very interesting is that the sorting technology itself is also very unique and based on microfluidic sorting, which is very gentle to the cells and leads to high viabilities and functionalities. So let's have a closer look on that sorting principle by starting looking at the closed cartridge. So you can see that the cartridge consists of 3 different chambers. We have an input chamber, also with a mixing propeller preventing from the cells settling down. And this is where your heterogeneous starting material goes into. And then we have a positive collection chamber where the target cells will end up, which had been sorted, and all the rest simply flows through into this negative collection chamber here. The heart of the system itself is actually the microchip at the bottom of the cartridge, which is also depicted here. It's actually only that small part you can see here, and just to get a better idea, I also included the size comparison with a match here. So we're really talking about a very small microchip, and to get a better understanding, it's important to zoom in. So here we can already see how it's implemented at the base of the cartridge and also connected with these channels here to the different chambers, the input, the positive, and the negative collection chamber. So let's zoom in further, and here we get an idea how the microchip itself really looks like. So the cells will enter in this hole here into the sorting channel where they are analyzed, and then a sort decision is made by the system. And if the cells are identified as target cells, they will very gently be redirected into the upper channel here, which leads to the positive collection chamber, and all the rest simply flows through into the negative collection chamber. And the sorting process is actually mediated by a micro valve, which is sitting here, and that can be opened via a magnetic pulse. So we can also zoom in to get a better understanding of this process here. So the cells coming from the left-hand side will just run straight ahead into the negative collection chamber. But if a target cell has been identified, a magnetic pulse is coming from a solenoid from the system, opens this valve here, which then redirects the cells into the upper compartment. We can also have a look on how the sorting process itself looks like for the whole of the cartridge, and therefore I also would like to show a short video that you can get a better understanding how the whole process works. So you can see the cartridge, and really the only thing coming from the instrument is actually sterile filtered air, which enters the cartridge at the bottom, running through an 0.1 micrometer filter inserted in the cartridge, and then entering the input chamber, again being sterile filtered with another 0.1 micrometer filter. Inside the input chamber, it builds up a very mild air pressure and then brings the cells through the microchip where the sorting takes place, and the positive sorted cells will end up here, and instead all the rest will simply flow through into the negative collection chamber. And this is how the sorting process then really looks on the microchip. Those cells are running one by one through the microchip channel here, passing the lasers, and as explained before, if a positive event has been identified, a magnetic pulse will open the microvalve, which very gently redirects the cells into the positive collection chamber. And yeah, that's the technical basis, the technical principle of the MACSQuant Tyto. And of course, we look also at different cell sorting applications in the regenerative medicine field in general. And the Tyto has been widely used for different applications also in the field which are connected to regenerative medicine. If we talk about stem cells, we see several applications around mesenchymal stem cells, neural stem cells, and also hematopoietic stem cells. But since these won't be the topic today, I just like to note that you can find more information about all these different applications here on our website, and also by following these links, which are provided also with a handout of the slides of this webinar. Today, I'd like to talk a little bit more about the induced pluripotent stem cells and also their derivatives, such as cardiomyocytes or neural progenitors. So let's have a first look on how a purification using the Tyto could look like when working, for example, with GFP-positive iPSCs in a research environment, right? So here you can see an analysis of the starting material. You see that in the input material, we have around 20% of GFP-positive cells, and just by passing them through the MACSQuant Tyto, we can enrich this population, purify this population to over 97%. And viability is not affected at all in these very sensitive, very fragile cell types. But obviously, it's not only about viability, it's also about functionality. And in this case, the cells had been replated and directly compared to different controls. And you can see that also the cells which had been sorted using the MACSQuant Tyto were adhering and adhering to the plates efficiently and also in a comparable manner as compared to the controls here, meaning that you could easily take them back into culture after the sorting process. We did more or less the same also for iPSC-derived cardiomyocytes. These were sorted on day 6 of the differentiation protocol. The sorting was based on our PSC-derived cardiomyocyte isolation kit with a positive and a negative marker. You can see in the starting material, we had a 40% purity of our target cells, which we again could enrich to 97% again, and a viability with over 90%, which of course is also a great result for these larger and also very fragile cell types. We took those also back into culture and also checked for specific markers of the cardiomyocytes, such as alpha-actinin. And as you can see here, we have a very pure and very viable result in terms of taking these cells back into culture, and they even started to contract within the first 24 hours after taking them back. And in the next part of my presentation, I'd like to focus a little bit more on the fact that the MACSQuant TytoCell Sorter can also be used in clinical settings. And again, why is that possible? So we have a very unique cell sorting system here because the sorting happens exclusively in the fully closed and sterile cartridge. There are no fluidics inside the instrument itself. And currently, the MACSQuant Tyto has already been approved for 10 different clinical trials, either phase 1 or phase 2 in different countries, such as US, such as Japan or Korea. And over 200 patient samples have already been sorted using the system. And another great benefit of it is that all consumables are not only available in research use only grade, but also in MACS-GMP grade. And that means that they come with a very extensive documentation. So you will receive an extensive documentation for everything that touches the cells, which would be obviously the MACS-GMP Tyto cartridge, but also our Tyto running buffer is available in MACS-GMP grade. And we also have a certain portfolio of fluorescent antibodies in MACS-GMP grade. And this will, of course, help with discussions with the regulatory authorities when translating from research settings into clinical settings, which is also possible that you use the research consumables in the beginning to set up your process, and then once you translate into the clinics, you simply switch to the GMP grade, and this helps a lot, as I said, translating your protocol into clinical environments. One example where this was done is coming from Japan, from the Center of iPS Cell Research and Application in Kyoto. And the group, also headed by Jun Takahashi, is working on a clinical trial utilizing iPSC-derived dopaminergic neurons in order to treat patients suffering from Parkinson's disease. It's really pioneering work that they are doing. So once asked in an interview on the platform RegMedNet concerning the question what the biggest challenge had been developing this therapy and bringing it to the clinical trial, Jun Takahashi answered that it was the sorting of the current positive cells which had been the most demanding step in that translation into the clinical trial. And I'd like to tell you a story about that. So in the beginning, they were working with conventional droplet sorters, so conventional cell sorters. We see them in the market. I mentioned before, they are open systems. They share tubings and fluidics, and you can exchange them, but it's very, very difficult and time-consuming and also expensive. And yeah, they had some problems with the system they were using. And so they were interested in bringing the MACSQuant Tyto into the clinical trial and performed a direct head-to-head comparison of the different cell sorters in their protocol, where they generated iPSC-derived neural progenitors. And they analyzed, after they sorted the cells and finished that differentiation protocol, they analyzed, of course, sorting performance, purity, and sorting yields, which had been comparable between the two systems. But they were, of course, also interested in survival, in cell numbers, and in functionality of the cells. And this is what I would also like to show here today, what had been done and how the results of this look like. So first, for a better understanding, this is the differentiation protocol which they are applying. And thanks again to Daisuke Doi from Saira, who kindly provided the data here I'm going to present. And what you can see here is that they worked with an established iPSC line, expanded the iPS seals, and then started the neural induction. And on day 12, 13 of their protocol, they need to purify the current positive cells via cell sorting, then go into post-sort differentiation for another 13 days when they perform QC. And if they are happy with the dopaminergic progenitors they were able to generate, the cells will be finalized for transplantation into the patients. And this is where the cell sorting step comes in. And what had been done is a direct head-to-head comparison. So cells were sorted using the conventional FACS droplet-based system, or the MACSQuant Tyto. And then on day 13 post-sorting, so the day of QC, the cells were analyzed in terms of how they grew and how the spheres, which are these cells formen, look like. And as you can see already by eye here, is that the spheres which had been formed by the cells sorted with the MACSQuant Tyto are much bigger in diameter compared to the cells which had been sorted using the FACS droplet sorter. This is significantly different and also leads to a higher overall cell yield after 13 days post-sorting, which is of course also very important for the researchers as they need quite an amount of cells for the transplantation. Another important fact is, of course, the functionality of the cells. So these cells will be transplanted into cells, and they also need to engraft there. And a way to analyze this in vitro is the neurite extension assay, which is an indicative of graft survival capacity. And you can see here already by eye again that there had been quite a difference between the area which had been covered by these neurite extensions between the FACS droplet sorter and the MACSQuant Tyto, which is also significantly different and higher in the Tyto sorted sample, which speaks for a very high graft survival capacity of these cells. Last but not least, it's all about time as well. The group needs 1.2 times 10 to the 7th cells to treat one patient, and using the droplet sorting system, they needed 18.5 hours of sorting procedure to end up with that amount of cells, and they were able to cut that down with a MACSQuant Tyto by 1/3 to 12 hours. And this is actually also utilizing our standard cartridge. In the meantime, we also released a high-speed cartridge, which runs at double the speed. So this could even further cut down here. So let's shortly sum up what is happening in Japan. So the MACSQuant TytoCell Sorter has been included into that clinical trial on treating Parkinson's disease using iPSC-derived dopaminergic neurons, and to date also several patients have been sorted successfully. If you're interested in more details concerning the story, there's also a poster summing up all the different essays and the results which had been presented by Daisuke Doi at the ISSCR in 2019. You can find it on our website and also by following this link which is provided here. And with that, we are coming to the end of our webinar. Again, thank you very much for your participation. I'd like to shortly sum up maybe the main take-home messages. So I hope we could give you some more insightful and useful knowledge about the solutions we offer for manufacturing and sorting of PSCs and their derivatives. On the one hand, the GMP-compliant Adherent stem cell manufacturing with the CliniMACS Prodigy. Obviously, also the iPSC and QC assays, which can be easily performed using our MACSQuant analyzers. And then, last but not least, if you're in need for purifying your cells for your protocols, the MACSQuant TytoCell Sorter is a good option to do that in a gentle and in a GMP-compliant way. And with that, I just need to shortly show our disclaimer here for a few seconds. But with that, I'd like to thank you very much for your attention. As we already mentioned, we are very welcome to directly reach out either to my colleague Chao or to me if you would like to discuss any certain topics in more detail. And with that, thank you very much.